All right, welcome everyone to another SNAC meeting, seminar of our seminar series here, organised with Max Shine and Joe Lizier at Sydney University. And this week we have a very special guest, Dr. Aurina Arnatkeviciute. How's that?
Good.
It was good. Who was one of, I co-supervised her PhD when I was back in Monash. She was extremely talented back then and has gone on to do even more amazing things since leaving her PhD, and she's now a research fellow at Monash University in the School of Psychology. And today she's going to talk to us about some joint work. It started as an early project from me chatting and having coffee with someone called Jeanette Tong back in the Monash days when we were both on fellowships, and then it kind of dried up, and then Aurina's revived it and made it her own and done some really cool work on linking genetic vulnerability for different psychiatric disorders to how we might go about assessing different drug targets for those disorders. So thanks for coming and joining us, Aurina. We're all looking forward to your talk.
Thanks for inviting. I'll start sharing my screen now. All good?
Yep. Okay.
Cool. Just move this here. That should be fine. So, hi, nice seeing you all. And today I'll be talking about this important general question in psychiatric genetics. So the question is what is the relationship between common genetic variation that can be identified through large-scale genome-wide association studies and the pharmacological treatments for psychiatric disorders. So over the last decade, there have been a large number of large-scale genome-wide association studies that have identified disease-associated genetic variations across psychiatric disorders. And one of the core goals of those GWAS studies initially was to inform the development of pharmacological treatments. And it was done on the assumption that if we select genes that are important in driving the risk for a disorder, then treatments that influence the function of these genes should mitigate the risk or alleviate the symptom expression for a particular disorder. However, in most cases, pharmacological treatments for psychiatric disorders, they have been developed without the direct genomic evidence, and it remains unclear whether the mechanisms of action in alleviating the symptoms relates to the actual genetic variation that is identified in the GWAS. It has been shown that drugs with direct genomic support, they move further along the drug development pipeline, and they also are more likely to be clinically successful. But actually, a very small fraction of treatment targets have been so far identified using genome-wide association studies. And psychiatric disorders also present a specific case where some of those disorders, such as autism, ADHD, and schizophrenia, they are regarded as neurodevelopmental conditions. So in this case, risk genes that may influence the brain development in ways that creates a vulnerability to illness may be different from the processes and mechanisms, and also sort of different in their nature, and also different in the time when they're acting from the mechanisms that influence the symptom onset and the severity. And these mechanisms would be the closer ones to actually being used as effective treatment targets. So in this case, we wanted to investigate this fundamental question and this common assumption. So do those mechanisms actually overlap? And to do that, we introduced methods to evaluate whether those genes implicated by GWAS are related to their drug targets. through independent bioinformatic datasets. So we used an array of approved treatments from the drug bank database and GWAS summary statistics for several disorders such as ADHD, schizophrenia, bipolar disorder, major depression, and also diabetes, for a comparison to a complex non-psychiatric disorder that has effective pharmacological management to date. And we constructed two sets of scores for each of the 2,155 genes in the drug bank database. So the first score quantified the involvement of each gene as a treatment target for the disorder. And the second score quantified the similarity of each of those genes to the variants identified in the GWAS. So in a treatment-based score, genes that are targeted by more specific drugs are assigned higher values. So in this example, if we look at the example here, for example, drug 1 is targets three genes, while drug 2 is targeting 5 genes. So genes that are targeted by more specific drugs, so in this case drug 1, will be assigned higher scores compared to a less specific drug. Drug 3, for example, is only targets 2 genes, so these get even higher scores. And gene weights across all drugs for disorder are then summed. Into this one single treatment-based score vector, and genes that are targeted by more drugs and more specific drugs then get higher scores compared to the rest. So in this example, gene 3 and gene 6 have been targeted by specific drugs and have been hit multiple times. So these are the highest scores that we get. So the second score quantifies gene similarity to GWAS-identified variants, and the first independent way of doing that is by positional mapping. So here each gene is assigned a score based on the effect sizes of SNPs that directly map to that gene or that are in linkage disequilibrium with it. So sort of they're co-inherited with the SNP of the effect. The other mapping method that we used based on protein interaction networks, and here we examined or sort of expanded the mapping to the first degree neighbors of our target genes. So in this example, we calculate how many of those neighbors of our target genes are implicated in a GWAS. So genes that have higher proportion of their neighbors involved in a GWAS get a higher score. So in this case, if we look at gene 3, 2 out of 3 of its neighbors are implicated in a GWAS. But if we look at gene 2, then only 1 out of 6 neighbors that this gene has is implicated in a GWAS. So therefore, gene 3 is scored higher than gene 2. And the third mapping method was based on brain EQTL mapping, which assigns higher scores to genes whose expression is influenced by the GWAS SNPs. And the fourth mapping method is based on regional gene expression data derived from the Allen Human Brain Atlas, which provides spatially comprehensive gene expression maps across hundreds of regions in the human brain across more than 20,000 genes. So to get that score for each gene, we first calculated co-expression between each pair of genes, which indicates which genes have similar spatial gene expression patterns. And then we assign higher scores to genes that have more similar expression patterns to GWAS implicated genes compared to the rest of the genes in the database. So as a result, for each of the more than 2,000 genes, we get those two sets of scores. So one is based on the treatment targets and one for each of the mapping methods based on the GWAS. And to quantify the correspondence between those scores, we calculate the inner product between the two unit normalized vectors, which creates a weighted similarity score. So as a result of this calculation, only genes with non-zero scores in both GWAS and treatment-based score vectors are contributing to this weighted similarity score. Another critical aspect is how do we evaluate whether the match that we get from those two vectors is significant or is it expected by chance. So to do that, we create a null distribution using random treatments. So basically we assign assign scores to genes based on randomly selected drugs. And then we create a null distribution and we assign each association a p-value associated with that. But I'll get into a bit more detail about this null assessment in a couple of slides. So first, we looked whether GWAS for psychiatric disorders matched their treatment targets for each individual mapping method. So the goal was to see which of the 4 mapping methods was the most informative in general. And overall, the results show that the degree of correspondence between treatment targets and GWAS findings across mapping methods and bioinformatic data sets, it is really low. So across all the mapping methods, no associations were identified for major depression, ADHD, and schizophrenia. As you can see here, those are down at the bottom. And in these plots, the significance of the association is on the vertical axis and each disorder is on the horizontal axis. If we look at each individual method in isolation, we see that for PPI network-based mapping, it was the most informative for 2 disorders, so diabetes and bipolar disorder. We see the associations that actually exceed the null expectation. Regional gene expression mapping as a method, it also identified associations for bipolar disorder. SNP position-based mapping showed associations for the same 2 disorders, bipolar and diabetes, but at lower significance thresholds. While EQTL-based mapping did not exceed null expectation for any of the disorders. So here we go back to the significance testing, and we aimed to address this question and create a sort of realistic way to assess the significance of those findings. So if we expect the association between GWAS hits and our treatment targets for a disorder, we expect that these hits will be more tightly matched to respective treatment targets compared to any other drugs. So we want our GWAS for, let's say, schizophrenia to be matched to schizophrenia drugs, but not to other drugs in general. So we want that association to be specific. And to evaluate that, we compare the degree of correspondence to selections of random treatments. And in this case, we can ask a question, do genes implicated in a GWAS match treatment targets better than any other random drugs would? And also we can specify this question from a slightly different perspective and constrain our null selection a bit more. So if we, for example, limit our random drug selection to only psychiatric drugs, we can then ask a question, do genes implicated in a GWAS match treatment targets better than other psychiatric drugs would? So it's a more constrained null testing. And in this example, it's clearly shown in the null distributions for diabetes. So under the random drug null, so the gray distribution here, we can see that this association in the empirical data is significant, but is much less significant compared to the random psychiatric drug null, which provides a much more narrow distribution compared to the all drugs in the database. So this actually happens because treatment targets for diabetes, they're much less similar to psychiatric treatment targets in general compared to the rest of the database. So this allows us to sort of tailor the question based on based on the disorder of interest, for example. So if we investigate non-psychiatric disorders, so we might want to remove psychiatric disorders from our null drug assessment, sort of not to bias the results based on psychiatric treatments. So next, we looked at pairwise correspondence between all disorders and mapping methods. So this was an exploratory analysis that allows us to evaluate if treatment targets for a particular disorder match genes involved in another disorder, and it could identify those cross-disorder associations and also label high-level repurposing opportunities. So for example, maybe drugs for a bipolar disorder match GWAS hits for depression. So we would see that association. So in this plot, we see a matrix where different disorder GWASs are represented as columns, and GWAS mapping methods are represented as rows. So for each of the combination for a disorder and treatment mapping method, we can evaluate this correspondence. So in this case, when we look at the plot, each of the bars represents a weighted similarity score that we talked about before, and the color represents the significance of the identified association compared to the null distribution, which is also plotted as a distribution in the plot. So considering a high number of multiple comparisons in this case, none of the associations actually are statistically significant after correcting for multiple comparisons. But in addition to previously shown associations for diabetes and bipolar disorder, the strongest links that we identified in between disorders is between bipolar disorder, major depression, and schizophrenia. And these are the three disorders that are phenotypically related to some degree, and they also show genetic relatedness between each other. So if we look specifically, treatment targets for bipolar disorder demonstrate correspondence with the genes implicated in both major depression and schizophrenia GWAS through PPI and SNP position-based mapping. And similarly to that, treatment targets for schizophrenia are also linked to genes implicated in GWAS for major depression, while all other combinations of disorder and mapping method pairs do not exceed null expectation even at the lowest significance thresholds. So this basically provides us some grounds to think that these relationships between phenotype and genetic similarity, they also might translate to the mechanisms and relationship between the mechanisms identified through GWAS and the treatment targets. In addition to the four measures that I have been describing before, we also explored how different data processing options could impact this mapping. So for example, we can use different density of the PPI network. So if we choose only to include associations in the PPI network that have the highest confidence, or we can include more and more, so increasingly more links within the PPI network to identify the neighbors of our genes of interest. We also included EQTLs for different tissue types, such as blood, liver, and pancreas, under the assumption that they might be relevant for identifying associations for diabetes, for example. We also included chromatin interactions for different mapping methods, and across 2 developmental time points and 2 cell types within the brain. And in these plots, colors represent the individual type of the measure. And also, in addition to individual measures, we evaluated whether a combination of different mapping methods, just a linear combination of them, can outperform individual measures as a single instance. So in this case, the strength of the association is on this horizontal axis, and all the mapping methods are on the vertical axis. So as we can see here, none of the mapping methods and their iterations provide a significant correspondence for ADHD and major depression. And significant associations in this case would cross the second dotted line in the plot. The example of schizophrenia shows that aggregating information across multiple measures can improve the correspondence. So meaning that information derived from different sources can complement each other and act in a complementary way for informing drug targets. And in most cases, if we look at bipolar and diabetes separately, we see that a range of PPI-based measures are the most informative for these disorders. And it suggests that potentially indirect interactions through gene products might be involved in linking the mechanisms of genetic risk for disorder and the mechanism of pharmacological treatment for those disorders. So just to sum up, here we have investigated the correspondence between current treatment targets and GWAS-implicated genes using different bioinformatic data sets. And this was to test this underlying hypothesis and common assumption in the GWAS research that identifying the risk variance for disorder would sort of inform the development of drug targets for complex disorders. And it's sort of one of the backbones of what the whole field of GWAS was based upon. So we really wanted to see whether these mappings are actually correspond to each other. And in this case, we see that this common assumption in psychiatric research might be a bit further from the truth than everyone hoped. And it might be that for those complex disorders, that mechanisms that actually predispose someone for developing a disorder might be different from the symptomatic management that is needed when a person develops a disorder eventually. So here we show that drug targets for most disorders lack the correspondence to the genetic origins that are identified in the GWAS. And also in some cases, we show that incorporating independent information from other databases, such as protein interaction networks, can reveal the links between those two mechanisms. Through pairwise comparisons, we show that genetic relationships between bipolar disorder, schizophrenia, and major depression, they tend to extend and translate into pharmacological treatments. And investigation across a range of different processing options consistently showed that PPI-based measures were the most informative, at least for 2 of our 5 disorders that we investigated, suggesting that interactions through gene products might be involved in the mechanism of pharmacological treatment action. So the overall, however, the overall correspondence between GWAS and treatment targets is relatively low, and this suggests that the mechanisms driving the genetic risk and the symptom expression might be different, especially for psychiatric drugs that we see here. So some novel approaches might be needed and important to understand the actual important information in treating psychiatric disorders. Yes, so here I will just like to thank my collaborators and supervisors for this project. So Mark Belgrove and Ben Fulcher, and also everyone else who was involved, including Jeanette, Kang, and Alex that Ben mentioned before. And there is a preprint if anyone is interested to read through in a bit more detail. And that's the end. Thank you.